Hi, again. I'm Steve Russell, consultant hematologist and professor of molecular medicine at Mayo Clinic. And before I hand over to Bev Davidson at the end of this meeting, I'd just like to say what a privilege it was to be elected and to serve as president of the American Society for Gene and Cell Therapy over the past year. I do think we're at a historic juncture at the moment for the field of medicine. I think we're going to look back on this and we're going to see it as the time where we shifted from long-term therapies for chronic diseases to one and done genetic and cellular therapies. And I think we can all see that happening. It's very exciting. It's been a roller coaster year. We've had the pandemic and we've seen messenger RNA nanoparticles and recombinant adenoviruses coming to the rescue on the vaccine front. We've had new drug approvals this year, particularly in the CAR T area. We've other drugs, several of them moving closer to approvals. And we've had some drug approvals delayed or set back because of toxic events. Or because of the need for durability questions to be addressed. The underpinning technologies have been advancing very fast. And we got our first Nobel Prize awarded to Jennifer Doudna and Emmanuelle Charpentier. And, hopefully that's the first of many Nobel Prizes for the field, but you absolutely must watch it if you didn't see it, the fireside chat with Dr. Doudna. She is a truly inspirational leader, and it's a great milestone for the field. Industry involvement in the field is continuing to grow, and the society this year transitioned to independent management. With our own executive director, David Barrett, leading the charge. The changeover was seamless and he's a pleasure to work with. As far as this meeting is concerned, we had 6,844 people registered, which is considerably higher than we've ever had before. There were over 900 abstracts submitted. There have been 150 sessions, and all the oral and poster presentations have been outstanding. We've had problems with the virtual platform for the first couple of days, kind of reminiscent of the Colonial Pipeline hack that's been going on. But, the ASGCT staff have worked around the clock with the Inovia staff to try and address the problem. And I think we're back on top of it now. There'll be a kappa, obviously, and we'll make sure that we don't have a similar crisis in the future. But overall, it's been a great meeting, and I think it's great that the content is good. It's all been recorded, and it's going to be available for at least 30 days, more likely 60 days. So you can go back and you can watch anything that you missed, and you can rewatch anything you want to. I'd like to say a quick thank you to a few people, David Barrett and his staff, for all the hard work they put into this meeting. The program committee, the abstract review committees, all of the standing scientific committees, the board of directors, all of the speakers and poster presenters, and the attendees. So thank you very much to everyone. I think it's been a successful meeting up to this point. Now, I'd like to switch to the symposium and introduce Dr. Robert Martuza. Dr. Martuza is a professor of neuroscience at Harvard Medical School, and he's the chief emeritus of neurosurgery at Mass General Hospital. He's also a member of the National Academy of Medicine. So apart from being a brilliant neurosurgeon and unraveling the genetics of neurofibromatosis, he published a pivotal study in Science in 1991 that really had a seismic impact on the field of oncolytic virotherapy. And in that paper, he used a TK-deleted herpes simplex virus as therapy in a mouse glioma model. And that was really the starting gun for the whole now vibrant field of oncolytic virotherapy, in which Dr. Martuza has continued to excel and lead, and for which, in fact, he was granted the prestigious Golden Virus Award by the International Oncolytic Virotherapy community in 2017. Without further ado, Dr. Martuza's title today is Oncolytic Immunovirotherapy, Uniting Genetic Engineering, Virology, Immunology, and Cancer Research.
Well, good afternoon, and I feel very honored to give this talk. I want to thank Steve Russell and the organizing committee for inviting me to do this. Up top is my contact information. If you have questions afterwards, down below are my disclosures. I won't read them, but you can look at them. And this is a somewhat complex title that I chose, Oncolytic Immunovirotherapy, Uniting Genetic Engineering, Virology, Immunology, and Cancer Research. And the reason for this title is that these are biologic areas that have been of great interest recently, as noted by Nobel Prizes given out in the last few years for cancer therapy, immune regulation, viruses, and genome editing by the people that are shown here. And in fact, oncolytic virotherapy combines all of these, and I will show you this over the course of the next number of minutes. Now, this research began in neurosurgery, which is unusual for oncology. Usually, central nervous system tumors are the last place that drugs go. But the problem is we had a seemingly unsolvable problem, glioblastoma, as shown here on this scan. And over the last number of decades, only a few compounds have been approved, and they have had marginal efficacy. Consequently, glioblastoma has a short survival time, about 15 months median survival, but its cellular characteristics are similar to many other cancers. It has intratumoral cellular and genomic heterogeneity, cancer stem cells, mitotic activity, diffuse infiltration, necrosis, angiogenesis, and it's highly immunosuppressive with impaired MHC1, with downregulation of important immune cells, and with immunosuppressive factor expression and recruitment of Tregs. So this was a big challenge. And I had noticed that the things that cause cancer were the same things that cure cancer. So for example, chemicals. Nitrosourea, if you give it to an animal, will cause gliomas and other tumors. And nitrosoureas like BCNU were the first things that we were giving people back in the 1980s when this began. Radiation will cause tumors such as gliomas, meningiomas, and others. And radiation is one of the first things that we give. And why? Because both these chemicals and the radiation alter DNA, but in non-specific ways. Viruses cause cancer, and there are multiple examples of this, HPV, various leukemia viruses, etc. But no one was using viruses back in the 1980s when we began this work. And that seemed to be an approach that was worth looking at. Now, as an academic, the first thing you do is look at the literature, and I call this Take a Walk on the Wild Side. This is a song by Lou Reed in 1972. And there were spontaneous remissions of various cancers from people who got a viral infection. And in fact, viruses, wild-type viruses, were given to people and various remissions were seen. The problem is these wild-type viruses had their own toxicities, including in sometimes fatal toxicity, and they were abandoned. Surgery, chemotherapy, radiotherapy became the mainstream therapies for cancer. But in the 1980s, we were in an era of genetic engineering, and the possibility of getting a virus to specifically grow in cancer cells, as is done in oncolytic virotherapy, as shown here, where the virus goes into a cancer cell, makes multiple copies, replicates, and goes on to kill other cancer cells, whereas in the normal cell would have no effect. This was a real possibility. And evolutionarily, viruses have done this, of course, and are very specific. Poliovirus you ingest, it goes through your circulation, but it only kills off the anterior horn cells of the spinal cord, not other neurons, not other cells in the body. So we thought we could engineer a virus that would specifically kill cancer cells. And we looked at various viruses, both RNA and DNA. And for reasons I'll mention to you in a moment, settled on herpes simplex virus type 1, which is a large DNA virus. With retroviruses, we were concerned about inefficiency, small package size, instability, secondary events due to integration, lack of effective antivirals. And of course, we were in the rampant HIV/AIDS era, and the FDA, what we thought, would never approve this. So we settled on herpes simplex because DNA compared to RNA is very stable. It's a large size virus, so you can make multiple genetic engineered mutations in it. It does not integrate into the genome. It's known to spread in the brain when we're trying to treat a brain tumor. It comes in multiple types, or as we call them, flavors, replicative, defective, latent, and antiviral drugs were available. And this was important then because we were concerned that this first time it was being used, we might have to turn it off. And things like acyclovir and valcyclovir will turn off a herpes virus. I think it's still an important concept that as we look at more aggressive viruses being made. So this is really a life-size oil painting of the first viruses genetically engineered being used for oncolysis. This is Jim Marker, who will come up later in the talk. This is me at a much younger age without the gray hair that I have now. And Jim is actually counting a virus called DLSPTK that was made by my colleague, Don Cohen. So we published this first paper in Science, 1991, and we could show that in a human glioma implanted in an athymic mouse, that increasing doses of the virus would give increasing levels of survival. And this dose-dependent nature suggested efficacy and something that is worthy of pursuit. And we suggested that a virus could be genetically engineered to kill cancer cells without harming normal tissue, in this case, the brain. And we proposed mechanisms including direct cell killing by the virus, production of new antigens on the tumor to induce immunologic rejection, and that genetically engineered viruses are worthy of further exploration as a means of therapy for tumors resistant to currently available treatments, such as glioblastoma, but also other cancers. We then entered what I call the dark ages, because when we applied for grants at the NCI, they said, well, glioblastoma is not really a cancer, it doesn't metastasize. In this very meeting, I gave a talk at one of the first meetings, and people said, well, it's not really gene therapy because you're not integrating something for further expression later. And when we approached pharma, they said it was too risky. They did not want to put their money into it. They've now ended up putting tons of money into it, as I'll show you later. Ultimately, confirmation by other groups is what really changed the field. Chambers in 1995 using a herpes virus showed that oncolysis worked, and Bishop et al. Using an adenovirus in 1996 in Science showed that it could work also. And this opened the field for many other investigators worldwide to get into the area. Now, in the meantime, we were making another virus. The first virus, DLSPTK, the basic concept was to take out thymidine kinase and use the fact that cancer cells had high levels of TK, brain cells did not, and so you could selectively kill cancer cells. The problem is you took out the TK gene, and therefore it was no longer sensitive to acyclovir or valcyclovir. It also still had some toxicity to brain, so we never took that to clinical trial. But we next made this virus called G207. You'll hear a bit about this as we go on. And this basically had two widely spaced mutations in this herpes genome. One in gamma 34.5, which is the major site for neurovirulence. So you take it out and you don't get herpes encephalitis anymore. And this was shown by Joni Chow and Bernard Roisman. And the other was ICP6, which is basically an analog of ribonucleotide reductase. And the deletion of this selects for replication and dividing cells. And by having widely spaced mutations, you would not get a reversion to wild type of any concern. So this was in 1995 in Nature Medicine. We went on in the next 5 years to do the first clinical trial of a herpes virus in the United States. And we could show in the patients that were injected, there was no evidence of herpes encephalitis, despite injecting this directly into the brain tumor. There were about a third of the patients had MRI treatment responses with decrease in the enhancing volume. There was one possible histologic complete response and 2 long-term survivors. And these were in biopsy-proven recurrent glioblastoma. And chronic toxicity did not occur in the long-term survivors. But it was also clear that one viral injection was not curative. patients did go on to die despite these remissions in some patients. So the next concept that we developed was the idea of in situ vaccination, which is really the start of oncolytic immunovirotherapy. And the idea was that if you had tumors on both sides of an animal, you could inject one tumor and get oncolysis and an oncolytic response. But in the opposite tumor, there would be no virus, but you'd get an immune response. And that is shown here, the inoculated tumor, the control, and the virus injected. And the non-inoculated tumor on the opposite side, tumor volume shown here, the control, and you get this response, which we could show the virus had to grow in the tumor on this side. If you put it in the skin near the tumor, it wouldn't work. We could show the tumor antigen involved that the immunocytes were responding to, and we could show that lymphocytes were necessary. And if you look at the paper, you can see all of this. Now, improving oncolytic immunovirotherapy was shown in two other manuscripts, one where we expressed IL-12 and the other where we expressed GM-CSF. And basically, that's as follows. If you look here, this is the inoculated tumor and the non-inoculated tumor. And basically, this is the control volume. This is the volume of a virus without IL-12. And this is the better response with IL-12 being expressed, both in the inoculated tumor and on the opposite side in the non-inoculated tumor. And this is a dose response using a GM-CSF expressing vector, where you can see the higher expression of GM-CSF is associated with decreased tumor volume in both the inoculated and the non-inoculated tumor. And GM-CSF, of course, is what is currently in TVEC, which we'll get to later. The other thing that we showed in that same time period was, one, the effect of prior exposure of herpes simplex. So basically, if you took animals, inoculated them in the foot pad, got an antibody response, you could show that HSV seropositivity did not deleteriously affect the efficacy of intratumoral therapy. But more importantly, probably, we could show that if you gave multiple inoculations, in this case, for example, there are 6 inoculations, as shown by these arrows here, of a 10-fold less virus than one inoculation, which is this line here. So this is a single inoculation, and these are the multiple inoculations in this animal at 10-fold less, 6 doses. You get a much more marked response. And this will come to play, as you'll see, in one of the trials that Tomoki Todo, who was a postdoc at the time, did with G47 Delta later on. And these are the survival curves. But the question then became, well, how do you overcome virus-induced immune evasion? Any virus, herpes being one of them, but also others, have ways of avoiding the immune system. And with herpes, there were particular ways that were done. And I'll show you the next virus that was made in a moment. But this virus that I'm talking about now, G207, was tried in all other adult glioblastoma trials. And this is a summary, basically. 50% of the patients had radiographic responses, pretty much the same as in our first trial. There were significant immune infiltration of CD4s and CD8s, again, very important. And patients could be treated sequentially multiple times without toxicity. Resistance did not occur, and retreated patients had radiographic responses after each new treatment. And the survival was superior to published data in the patient population. This was compiled data from Jim Markert plus the trial that we did initially. Now, very recently, just last month, April, Jim Markert's group down at Duke, led by Friedman and Yancy Gillespie, used G207 in pediatric high-grade glioma patients. They had 12 patients, this was in the New England Journal of Medicine, with recurrent malignant supratentorial gliomas that progressed after all of the treatments. They gave a one-time treatment of a catheter infusion over 6 hours of this amount of virus, and they compared it to historical controls of 5.6 months. Now, the interesting thing is the median overall survival was double, but possibly more important, and this would have to be borne out in a larger study, those patients that were seronegative had a much longer survival, about 3 times the normal survival. And so this may be a discriminating feature. Other people, such as Eva Galanis, had showed other discriminating features with their measles studies, including various ISG and interferon responses. But in this study, the delayed response occurred over several months, and this is important and will be seen also in a study that Tomoki Todo did. And there were significant increases in these various tumor-infiltrating lymphocytes, and now they're going ahead with a phase 2 study, much larger, which will probably be multi-centered and more important. And this is an example of the delayed response that one gets after oncolytic virus therapy in glioblastoma. This is before treatment. You can see an enhancing tumor here, and you can see the edema here. This is at 1 month, 5 months, 9 months. You're seeing various cysts develop. And at 24 months, a much smaller tumor, much more cystic degeneration, and a lot less edema in this patient. And if you look at the patients who ended up being biopsied, this is 4 different patients, patient 1, 6, 9, and 10. Before treatment, there was really minimal or no lymphocytic response. After treatment, there's a significant lymphocytic response, even as long as 9 months later after a single viral dose. And this concept of immune response in a delayed fashion, correlating with the MRI delayed response, is something that's being seen over and over again. So as I mentioned to you, viruses have ways of avoiding the immune system. And the next virus that was made is called G47 Delta. It's basically G207 with an additional mutation in ICP47. Now, ICP47 inhibits MHC1 class presentation. So again, it will get over some of this viral blocking of the immune system. So the deletion should overcome this, thus improving viral therapy. It also leads to an early expression of US11, complementing the gamma 34.5 loss, which enhances viral growth and increases oncolytic viral activity. And this was initially described by Ian Moore. So unknown to us at the time G47 Delta was made is one additional feature, and that is its effect in cancer stem cells. So glioblastoma-initiating cells, or glioblastoma sphere cells, were first described by Singh and Dirks in 2003. And basically, instead of growing cells in a monolayer, you grow them as what are equivalent to neurosphere cultures. And when you do that, you end up with these so-called stem-like cancer cells, which form tumors at 1,000 cells less than those that are normally grown in a monolayer, and that's shown down here. So what we found out was that G47 Delta replicates in these glioblastoma stem-like cells, whereas the original virus, G207, is not very good at it. Here's the titer after time in vitro. G207, basically the same. This is MGG8. This is a human glioblastoma stem cell-like culture. G47, as you can see, grows to a higher titer. The same in this human glioblastoma stem-like cell culture. G207 goes down, G47 Delta goes up. And you can see that in a GFP expressing G47 Delta, where these neurospheres are shown at 24 hours and then at 48 hours. Now, we didn't know this when the virus was first made, so this was really serendipity, but very important, I think, for curing various types of cancer. And we didn't know at the time this was shown what the cause of this was. But Cole Peters, who was a postdoc in the lab in an article in 2018, demonstrated that gamma 34.5 deletion in G207 allows growth in monolayer glioblastoma cells, but not in the glioblastoma stem cells, due to a block during translation of true late HSV genes in the stem cells. The G47 Delta mutation of ICP47 overcomes this block. And so that's the reason this works. And so for that reason, G47 Delta appears to be better than G207. Now, I mentioned Tomoki Todo, who started out as a postdoc in our lab. He's now a professor at University of Tokyo. And he finished his first phase of a clinical trial, which is a phase 1/2 trial, stereotactic intratumoral inoculation in recurrent glioblastoma patients refractory to other therapies, doses in the 10 to the 8 to 10 to the 9 range. And interestingly, doses were given at a monthly time period for up to 6 months. So again, the 6 times treatment, just as I showed you in that preclinical study, being better than a one-time treatment. And the 1-year survival in this group went to 92% versus 15% in the historical group. Very impressive. And this is an example of a scan in a patient, the initial tumor here, and then finally what appeared to be a complete response. So this virus in Japan, G47 Delta, in 2016 was considered as a fast-track breakthrough therapy. In 2017 was given orphan drug status. And in 2021, Daiichi Sankyo submitted a filing to what is the Japanese equivalent of our FDA. That will likely go through and will be approved by this Japanese equivalent of the FDA within about a month or so, and will be useful for glioblastoma patients. Now, let's step back a little bit and look at the generalities of this immune response. Tumors die by non-inflammatory, non-adjuvant apoptosis. Oncolytic viruses, on the other hand, can induce inflammatory cell death. Tumors have defective danger signal pathways, PAMPs, DAMPs. Oncolytic viruses can induce pro-inflammatory DAMPs and PAMPs. Tumors induce immune tolerance. Oncolytic viruses can break tolerance, elicit innate and adaptive immune responses. Tumor-associated macrophages are programmed to promote tumor suppressor T cells. These are so-called M2 macrophages. Oncolytic viruses can reprogram the tumor microenvironment, including the tumor-associated macrophages, into an M1 phenotype, which is anti-tumor. So to study these various immune responses, we needed an immune competent model. And fortunately, Maramoto and Inderverma had published one in 2009, using what are called mouse 005 GSCs. And you can isolate these from the tumor and get a neurosphere and then get gliomas, which are very invasive and grow in C57 black mice, which are immune competent. Now, the hallmark of glioblastoma stem cells in these 005 tumors is they can self-renew. They express stem cell markers such as CD33. They're highly tumorigenic. They're highly invasive, just like human glioblastoma, and they're poorly immunogenic. Here's an MHC1 assay. You can see very low level induced by interferon to show a higher level. MHC2, nothing and no interferon induction. And PDL1, low but induced by interferon. So this is a very good model for studying immunosuppressed tumors. And basically, it's a very resistant model to radiation and chemotherapy, importantly, again, like human glioblastoma. Now, oncolytic viruses are ideal for delivering anti-cancer compounds that are toxic when given systemically. There are a number of these, but one of them is IL-12. IL-12 is anti-angiogenic. It's pro-inflammatory cytokine, promotes proliferation of activated T and NK cells, stimulates Th1 differentiation, induces interferon gamma production. It's been shown to be effective in preclinical studies, but it's toxic to humans if given systemically. So it fell out of favor. But it's ideal for giving locally in the context of an oncolytic virus. So Tubachima was a postdoc in our lab and made this virus expressing IL-12, in this case, mouse IL-12, and made a comparable virus we call G47 delta empty, which doesn't have the IL-12 expression. And basically, in this 005 GSC tumor model, intracranially, IL-12 expression normalizes vasculature, decreases the Tregs, and increases survival. So you can see the vasculature here quantitatively, and you can actually see it very nicely on these histologic studies. PBS, G47 delta empty, and then much less vasculature in the IL-12 expressing G47 delta. And you can see also in terms of Treg expression, PBS, empty virus, 12 virus. And you can see it in terms of survival, where you get a slight increase with the empty virus, but a much bigger increase in some long-term survivors with the virus expressing mouse IL-12. And we could show, I don't have it up here, but if you go to the paper, you'll see it, anti-tumor activity dependent upon T cells. The next thing that came out in the whole cancer field was this idea of immune checkpoint blockade, basically taking the brakes off the immune system. So whenever you get an immune response, the body tries to respond by a braking mechanism. And there are many of them, but the two that were initially explored were CTLA-4 and PD-1, PD-L1. And antibodies were developed to be able to block these. And these have proven very effective, as I noted in the initial Nobel Prize award. Basically, in this paper by Lawrence in 2013, he could show that the tumors that were receptive and responsive to blockade of PD-1 or CTLA-4 were things like melanoma and lung adenocarcinoma, which he called hot tumors. Basically, they had a high mutation frequency, somatic mutation frequency, whereas other tumors like glioblastoma are cold tumors with much lower somatic mutation frequencies. And glioblastoma have low PD-1 expression, dominance of myeloid suppressive cells, multiple suppressive molecules, and T cell sequestration in the bone marrow. So these are hard to overcome, and all studies thus far with the immune checkpoint blockers in glioblastoma have basically been failures. We thought that basically this could be overcome. And so back in 2017, Dipankar Saha in our group was a postdoc and did this study. So basically this is G47 delta MIL12 in these 005 tumors, either by itself or with anti-PD-1 or anti-CTLA-4. And you could see that compared to the control, if you gave 1 dose of virus and 3 doses of the checkpoint blocker, you got a slight increase in survival. With either one alone, if you put them together, you got a slightly more increase in survival, but ultimately the animals died. CTLA-4 was a little bit more effective with virus alone. With the anti-CTLA-4, you got an increase in median survival. But again, if you put them together, about 90% of the animals still died. We thought that since these are two separate pathways of immune check blockade, if you took them both out and gave the virus, you might be able to enhance efficacy. And that, in fact, is the case. So here is the example with anti-PD-1 and anti-CTLA-4 combined relative to control. And here's the example if you give virus along with the 2 checkpoint blockers, you get a 90+% survival rate. And tumor rechallenge is 100%. They don't come back. If you inoculate tumors, they don't grow. And these are rechallenged brains here. This could then be shown with another model, CT2A. And again, the rechallenging showed that tumors cannot be put there. So there's immune memory from this as well. Now, the title of this paper was Macrophage Polarization Contributes to Glioblastoma Eradication by Combination Immunovirotherapy and Immune Checkpoint Blockade. And the reason for this is basically one of the things that was shown is that the macrophages went from M2 to M1 polarity. In other words, from pro-tumor to anti-tumor polarity. And treatment was associated with macrophage influx and this polarization to M1. This has since been confirmed in this paper by Kwan in this year, 2021. Macrophages mediate the anti-tumor effects of oncolytic virus 1716, a different virus in a different tumor, mammary tumors. Now, the reason this is important is up until now, people have thought it's only the lymphocytes that matter. It's clear that macrophages are very important in this process. And when we did depletion studies of CD4, CD8, or macrophages, we found that this is with no depletion, you get the survival that I showed you before. And if you deplete macrophages, either with BLZ945 or clodrosomes, you basically get a diminution and knock out this survival benefit. But the same is true if you knock out CD4 or CD8. So we're suggesting this is a very complex immune interaction between macrophages, CD4, CD8 cells, and all are probably very important in this process. So basically, what is said by this is that you can hopefully take glioblastoma, a immunologically cold tumor, use the virus to convert it to an immunologically hot tumor, and then use two different immune checkpoint inhibitors and take it up to a cure rate in the range of 50 to 90% or so, depending on the tumor type. Now, different tumors may require different immunomodulators, and this needs to be further studied. But I think it's going to be an important concept going forward. And people are, in fact, taking some of these immune modulators and putting them into the virus itself. Now, I will say it may not be the whole story. There was this paper by Matt Gromier this year, a couple months ago, showing that it's not just the mutational burden, but basically inflammatory gene expression probably has something to do with the patients who had a better response in their poliovirus study. Now, this is not just applied to glioblastoma. TVEC, which was approved by the FDA a few years ago and expresses GM-sphere, has been combined with immune checkpoint inhibitor therapy in metastatic melanoma. And here are two papers that show the improved efficacy of doing that. Paper by Rebus in Cell 2017 showed that the virus, TVEC plus immune checkpoint inhibitor, doubled the response rate relative to anti-PD-1 therapy alone, 62% versus 33%. And then another paper in the following year by Chesney in Journal of Clinical Oncology using TVEC and a different ICI inhibitor, anti-CTLA-4, showed that the virus plus checkpoint inhibitor doubled the objective response rate in, again, metastatic melanoma, and importantly, diminished the non-injected visceral lesions, 52% versus 23%. And this is that in situ vaccination effect that we had shown preclinically, and is called basically an abscopal effect, and very important probably going forward. And it's not just melanoma. Here's a paper from Ramalite from this year, a few months ago, showing that TVEC is also efficacious in B-cell lymphoma. TVEC was given in 13 patients. There were 11 objective responses, 6 of which were complete. 24 hours post-injection, the TVEC transcripts were detected in malignant and non-malignant cells of the injected lesion, but not the non-injected lesion. Again, showing it's not the virus going over to the other lesion, it's the virus giving an immune response affecting the other lesion, as we'd shown preclinically. So despite no virus in the non-injected lesions, there was an immunologic change in both the injected and non-injected lesions. There was interferon pathway activation, influx of natural killer cells, monocytes, dendritic cells, enrichment in cytotoxic T cells, and a decrease of regulatory T cells. So the efficacy and safety they felt in this lymphoma model, not model, but clinical trial of humans, was comparable with that reported in melanoma. So I think you're seeing now the expansion into other areas of cancer, and this will be continuing to go on. Now, this oncolytic virus platform is very rich for development. This is a partial list of other oncolytic viruses currently in clinical trial, and there are more than that. This is a partial list of other immune checkpoint inhibitors. I talked about PD-1 and CTLA-4, but there are a whole host of other checkpoint modulators. And I talked about GM-CSF and IL-12, but there are various other cytokines. And this is important because, again, if you start giving all of these systemically, you get toxicity, including myocarditis, which can be fatal, and various other itises or immune diseases. By putting them locally in the virus into the tumor, you can avoid these systemic toxicities. Now, what about other strategies? Intravenous therapy is very appealing to drug companies. There have been about 6 viruses that have been tried with intravenous therapy. The responses, except for one that I'll show you soon, have been relatively unremarkable. It's a complex problem for a variety of reasons. The other thing are novel virus constructs and synergies with other therapies, and many of these have been demonstrated. Here's probably the best demonstration of intravenous therapy that was done by Steve Russell and his colleagues at the Mayo Clinic. And this is using a measles virus that was engineered by Steve and his colleagues in a plasmacytoma. And you can see here this bone lesion in the skull before viral treatment, and then after viral treatment, it is gone. But this was one patient among a number who did not show similar responses. And the IV questions that remain, including the dilution of the virus once you put it in the bloodstream, non-specific absorption of the virus by platelets, by vascular cells, et cetera, innate and acquired immunity, which many people have to measles in this case, or herpes virus, et cetera. And then the issue of tissue specificity. It's a wide area of current study going on and a very important area. And I would emphasize this is a very active field of research. These are just 5 papers I picked of different types with different viruses from this past year alone. 1, transcriptional regulation of neurovirulence gene in herpes virus. This is the RQ-Neston study by Nino Kioka and his colleagues. Overcoming interferon resistance, again in herpes virus, designing an interferon-resistant virus by Kennedy and colleagues. Expressing immune checkpoint inhibitors by the virus. This is in vaccinia, but it's been done in other viruses such as herpes, et cetera. In this case, it's a PD-L1 expression by the virus. Involving other aspects of anti-tumor immunity, such as CAR T cells. This is a paper with vaccinia virus, but this is also being looked at with other viruses as well, and will be a very interesting area to study. And then finally, simply increasing OV replication and apoptotic tumor cell death. This is a paper in adenovirus. So again, a very active, rich area of study ongoing. And importantly, big pharma is finally getting involved. In 2015, Amgen announced FDA-approved T-VEC. 2016, Bristol-Myers Squibb signed a large agreement with Cioxis. In 2018, Merck made the biggest oncolytic virus bet since Amgen. 2019, Turnstone teamed up with Takeda in a $900 million deal to develop an oncolytic virus. So I think this will help really speed things up, taking the field forward. And in fact, as of a few weeks ago, there were 119 studies ongoing at 142 sites worldwide using oncolytic virus for cancer. Now, I think you have to realize some of these aren't going to work out. That's true for most cancer studies, but some of them will work out, and I wouldn't let the negatives derail the positive ones, because some of these, as I've shown you already, will be working out and take the field forward. So basically, what I hope to show you in this talk is the process of oncolytic viral development is very rich. It's iterative, multifaceted, synergistic with other therapies. By iterative, I mean that in mathematics, it means you take a function and repeatedly apply it over and over. Here it's bench to bedside, back to bench, back to bedside, et cetera. It's multifaceted. It started out as just oncolysis, but now I've shown you it's immunogenic. It can be tumor cell specific. It can kill stem cells, and it can be anti-angiogenic. It's not affected by hypoxia. And it can be synergistic with a whole host of various other cancer treatments. So this is a very rich area for further clinical development. Now, as I said to you, we've published the first paper in 1991. So this is now 30 years later. You might say that's a long time, but in reality, it's not that long. If you think about anti-angiogenesis, Judah Folkman described the first paper on tumor angiogenesis and anti-angiogenesis in 1971. It wasn't until 2004 that a drug that's commonly used now called Avastin was approved by the FDA. That's 33 years later. And it wasn't until 2012 that axitinib, an oral agent, was approved, and that's more than 40 years later. So this is within the range, and we already have T-VEC approved in the United States, an adenovirus approved in China, and G47 Delta about to be approved in the next month or so in Japan. So we're probably ahead of what happened with anti-angiogenesis and within the range of complex biologic treatments. I want to end by thanking a number of people, very important. This Don Cohen was the virologist involved in the initial paper in Science in '91. And since then, I've had a wonderful association with Sam Rabkin, a molecular virologist. Both of them are at Harvard, Sam at the MGH with me. And 4 former postdocs or neurosurgery residents who are now professors doing their own oncolytic studies. Nino Kioka, who I mentioned with the Neston virus, is chairman over at and professor over at Brigham and Women's Hospital at Harvard. Jim Markert, who was very involved with the initial clinical study and the other G207 study that was in the New England Journal recently, is professor and chairman at UAB. Tomoki Todo, who I mentioned to you, did the G47 Delta study was a postdoc in the lab, now is professor at University of Tokyo, and Masahiro Todo, who was a postdoc in the lab and did the initial studies on in situ vaccination and immunovirotherapy, and is now professor and chairman at Keio University. There are a number of other people I'm very thankful for in all of these studies. Some of them highlighted here I mentioned already. Others I just simply didn't have time to mention. So in ending, let me just say I thank you again for inviting me to give this talk, and I'm available to any questions that you have. Thank you very much.